Welcome to Nikolai's Genetics Lessons, and today we are going to talk about another case of gel electrophoresis. What's wrong with my gel? And here's a question from one of my subscribers. A friend did PCR and then loaded it on agarose gel. All samples were prepared in the same way. The PCR was identical with the same template, only primers differed. The same amount loaded on the gel, but 5 of them did something weird. Any idea what could be wrong with this? And we are talking about this part. So the reasons can be that the comp was set too deeply in agarose on the left hand side, gel tray not horizontal on pouring, and there is a sample spillover between the wells. So solution would be load smaller sample volume. And also the spillover sample is very near to the surface of the gel, so it's running quicker than the sample deep in the gel, especially if the gel is thicker on the LHS and is not well covered by running buffer. We also see here that the ladders on both sides got distorted. Possibly the DNA template has a lot of salt, and this was interfering with the uniform running of the samples, because the ladder bands close to the odd samples are being forced to run unevenly close to the amplified bands. And this is all for today. Subscribe, like, and see you in the next video. Goodbye.